in more than a century, since Louis Pasteur made very fundamental discoveries in understanding the germ theory of disease, here in Paris, we still struggle with disease, especially infectious diseases. Infectious diseases today claim millions of lives, and also infectious diseases are looming on the horizon to be among the top threats facing humanity in the years to come. So today I'm excited to be here to share for the first time with you a new vision for a new way of thinking about drugs, a way that goes to some of the very fundamental properties of biological systems, which is the ability to evolve. And the vision is to think of creating a new class of drugs that can learn some few tricks from biology, and the tricks that are laid in the ability of biological systems to evolve. But first, why is this an important problem? So, when Alexander Fleming discovered penicillin, we thought that this would mark the end of bacterial infections. But today we know that penicillin is in fact not active against many kinds of bacterial strains because the bacteria have figured out a way to escape from the drug. They become drug resistance. If you look at another totally different disease, malaria, the drug chloroquine, one of the most successful chemotherapeutics, was thought to mark the end of malaria. Malaria continues to threaten the lives of millions of people around the world, especially young children under the age of 5 years. Another drug, artemisinin, which was used by the Chinese for centuries, has now emerged as the first-line drug for the treatment of malaria. But in Southeast Asia, we are beginning to see the emergence of resistance against artemisinin. And this is true for many more drugs. Nearly every drug that has ever been developed by humans has faced resistance. If you think about antiviral drugs like antiretrovirals, all of those drugs have faced resistance. So we need to think of new ways to develop drugs because the threat of infectious diseases, it's real and it's going to get worse because of emerging pandemics. And it's very hard to know what's going to be next. We don't actually know what's going to be next, no matter what we do. But most unfortunately, is that when we are faced with a new disease, like Ebola, Zika, it takes us just too long to develop new drugs, and it's too expensive to develop new drugs also. And even when we do develop new drugs, the germs will always gain resistance. So we must develop new ways of thinking. And especially because when you think about the new pathogens that may emerge, we need a faster way of responding to new pathogens because we are not capable of doing that yet, not for any pathogen. And remember, even for those pathogens where we develop a drug, the pathogen can always gain resistance. So we need to stop this cat and mouse game of humans versus germs. Current drugs can't cope with this ability of germs or pathogens to evolve and to adapt to new challenges or drugs. And this is basically because there's a fundamental flaw in drugs. And that fundamental flaw is that drugs are static. They don't change. Once you design a drug, it doesn't change. It remains the same way you designed it. But pathogens, on the other hand, they change. When you design a drug to block one route for the pathogen figures out an alternative route to use instead of the route that you've blocked. So we need to develop drugs that are smart, because current drugs are not as smart as the pathogens are. Can we really make drugs that are smart? I really do believe that we can. And my research is focusing on how to specifically make drugs smart, make drugs to begin to have the property of evolution, which can enable the drugs themselves to begin to evolve and adapt to pathogens as the pathogen changes. The smart drugs I envision will work like this. So basically, when you have a pathogen that has a red region targeted by a drug that specifically binds to that red region, when the pathogen or germ changes the red region to a green, the drug itself will be able to change without a human intervening to target the new green path. So basically, when the pathogen mutates to resist the drug also mutates back to target the new version of the drug. And there's a very, very real reason, I believe, that this can be done. And the reason I do is because in the last few years, there's been an emergence of a new class of RNA molecule known as CRISPR. And CRISPR has been used by bacteria as a means for resisting viral infection. So CRISPR is an RNA molecule that enables certain bacteria to cut out viruses that infect those bacteria. But in the last few years, humans have figured out a way in which they can use CRISPR to cut any DNA of interest. So the DNA of interest might be a human gene, in which case the CRISPR is used to do genome editing in humans. But in another case, CRISPR can be used to develop a new class of therapeutics that target things like viruses by cutting specific viruses at specific places in their DNA. And I think there's a great potential for CRISPR as an antiviral therapeutic, where CRISPR is designed to target specific viruses based on their sequence. Now, in the last few decades, the ability to sequence DNA has become tremendously cheap, and it's going to get cheaper and cheaper. It's going to get also cheaper to do it in any part of the world. And this opens the new possibility when a new pandemic emerges to sequence the new virus causing the pandemic within a few days, if not hours. And from that sequence, if you can design a new drug, such as a CRISPR molecule that cuts the virus, then it means we can do it tremendously quickly than we would have using the conventional drugs. And of course, there's been evidence for this published in the scientific literature over the last 2 years, where CRISPR has been used to cut HIV out of infected cells. So this is really possible. But the problem is that, like any other drug that has been ever designed, HIV has figured out a way to escape a CRISPR that is targeted against it. And HIV can do this extremely fast. So in the experiments that were done in this study, the virus was able to do this within a few cycles of replication. So my work is looking ahead to see how to make a drug like an antiviral therapeutic based on CRISPR or any other kind of RNA molecule, how to make it smart enough that when the virus mutates or evolves, then the CRISPR itself will evolve and target the new strain of the virus. And so basically the way this works is that if I have an HIV virus in the genome, so an HIV provirus, which is basically the DNA version of the virus inserted in the human genome, I can target it with a specific CRISPR molecule that targets the blue region of that virus. But the virus can change. It can mutate over a few cycles of replication. It can mutate and replace that blue region that I'm targeting with a green region. But this new version of CRISPR will be able to evolve to now target the green region without human intervention. So all of this can happen in parallel with the virus in the human body. So the key difference with all the drugs that we've ever had is that if this happens in the current situation for any drug, we have to go back to the lab. First, we have to go and study why is the virus becoming resistant, and that can take years to do. And then secondly, we have to figure out a new drug to target the new version of the virus. And that can take a very long time and billions of dollars. So basically, this class of smart drugs are basically drugs that are able to evolve and adapt with a disease-causing organism of interest, such as HIV. But of course, it's not only important for infectious disease. We know that in cancer, for example, there's multiple chemotherapeutic drugs against cancer cells, but cancer cells can evolve resistance to most drugs. This is one of the biggest problems in cancer, because when you have a drug that targets the cancer cell can figure out how to become resistant within a very few cycles of replication of the cancer cell. So this could be widely applicable. But I think most importantly is that we need a new way of thinking about drugs. The one thing that has made biological systems successful is the ability to evolve, the ability to make changes in DNA or in the genetic material, which allows biological organisms to adapt to any new challenge. We need new drugs that can similarly adapt to this evolving nature of germs. Thank you.